Before Charles Darwin published his now famous evolutionary theories, his senior, Jean-Baptiste Lamarck, published his own ideas. See, Darwin wasn't the only guy studying evolution. There was a lot of people talking about evolution, and Lamarck was one of them. He argued that evolution occurs through what he referred to as an adaptive force. What he was saying is that organisms will alter their behavior to overcome environmental challenges, and this causes their biology to adapt accordingly. Like what a classic example of Lamarckian inheritance, which the future offspring would inherit, would be like a giraffe, right? So he's just saying the ancestor to the giraffe was some kind of horse-like creature, and the most nutritious, the most abundant food was above its reach. So this ancestor to the giraffe would stretch its neck to try to reach the food, and if it was successful, that now stretched neck would be inherited. So over the generations, the necks got a little bit longer and a little bit longer because these giraffes over the generations just kept stretching and stretching and stretching their necks until they could finally reach these large amounts of abundant food. So what he's literally saying here is that your children can inherit changes to their biology based on your behavior. Now, this has been thoroughly rejected by the scientific community. This is not how evolution works, right? Darwin, with his ideas about, natural selection, sexual selection, that's how evolution works, right? Well, kind of. Lamarckian inheritance has kind of made a resurgence recently, where we now are taking seriously a lot of the ideas that he was putting out there about this kind of adaptive force, but we're making one major modification to what he was saying. It's not exactly like this. A better way to describe the way it really works is that inheritance has layers, okay? So our understanding of inherited genetic predispositions has grown more and more nuanced, and now we understand there's a hierarchy of influence. So genetics is kind of like an onion, it has layers. So at the bottom layer, we know that genes predispose traits. Above that, we know about polygenic inheritance. So it's not that single genes predispose traits, it's actually that multiple genes predispose a single trait. On top of that, we know that genes are not your destiny. Just because it's in your DNA, that doesn't mean it's going to happen. The environment has to trigger these genetic predispositions. So you have environmental triggers of these multiple genes that could then result in a trait, right? Another layer on top of that is where we get to the epigenome. So epigenetics, this thing we call epigenetics, can actually alter those predispositions, and epigenetics can come from the environment. So the environment can alter your epigenome, the epigenome can then alter your genetic predispositions, and the environment then would not be able to trigger those predispositions, which are represented across multiple genes. So like I was saying in the previous video, this is a lot more complex than a lot of people understand. And I'm still oversimplifying it to a large degree, but this is the basic idea that there are these layers of influence to genetic inheritance. So when we talk about epigenetics, we're just talking about that how DNA can be modified by the environment, basically. And one of the most basic ways that the environment can modify your genome is through DNA methylation. This is the best understood epigenetic influence. So DNA methylation involves modifying DNA by altering the nucleotide base cytosine. So here you've got cytosine, and what we call it methylation because what's happening is a specific ethyl molecule is becoming attached. So now it's methylated cytosine. And genes containing methylated cytosine don't work. So the gene is basically just deactivated. This gene can no longer be copied. It can no longer be used to produce proteins. It's turned off for all intents and purposes. This methylation is performed by enzymes that you find mostly in the central nervous system, especially during early development. So this methylation I'm describing seems to have the most substantial impact on those most important early developmental stages. So that's what we're talking about here. Epigenetics, the study of heritable changes on how genes are expressed, caused by mechanisms other than changes to the DNA code. So epi basically means above, so epigenetics is like an influence that is above genetics. And basically, epigenetics refers to, in very simple terms, how genes can be turned on or turned off by the environment. This functionally allows organisms to adapt more rapidly to your environment. Like if you're born into an environment that is forcing you to be a certain way, your genes can quickly update, like quickly correct themselves to be more well suited for that environment. So DNA methylation is, one of those primary epigenetic mechanisms, but there is more to it than just methylation. But just to say a couple more things about that real quick, what's doing this methylation is called methyltransferases. So DNA methyltransferases, or DNMTs. These are those enzymes that perform the cytosine methylation. They initiate the transfer of that methyl group molecule. And we've got different kinds of DNMTs in mammals, like DNMT1 maintains methylation state across replication. So this is how children can inherit their parents' methylated traits. And DNMT3 initiates new instances of methylation. So this is what's doing the alterations. This is what's like deactivating the genes. So together, these genes, these DNMTs, can disable parts of an individual's DNA and prevent that part from functioning in the future. Here you've got your little DNMT doing its job, applying a methyl group molecule to this gene right here. Now, there's all kinds of things that can trigger methylation. DNA methylation will occur if DNMT3s are triggered by the presence of certain environmental agents. And we've done a little bit of research on this, and we've found a few things. Like we know, for example, diet can trigger it, hormones, stress, drugs, physical activity, disease, pollution, and there's a lot of other things too. But these kinds of environmental triggers can cause a wide range of methylation that can result in unpredictable developmental effects. So through DNMT3s, your DNA is being modified, like how your genome is being read and used is being modified by the environment. So DNA methyltransferases and methylation and all this is one of the big mechanisms. But there is another important mechanism, and that is histone, that involves histones. So histones, this is an abstract depiction of a histone, but histones, what they do is it's like a core of protein that DNA wraps itself around. It's what gives chromosomes their shape. Like chromosomes are these tightly wrapped and packed segments of DNA, and histones are responsible for that. So histones are important. To fit those 3 billion nucleotide base pairs of human DNA into our cells, we need these histones. They're very important. But how the histones are arranged can actually have an epigenetic influence. It can prevent certain genes from being read and copied and passed on to offspring. So like gene expression, DNA replication, and the creation of certain cell types can literally be deactivated depending on the arrangement of the histones. So histone modification is another mechanism through which we can influence the epigenome. So this refers to how the arrangement and functioning of histones can influence how genes are read. Two common methods that involve enzymes to add or remove specific molecules from the tails of histones include histone acetyltransferases, or HATs. These attach acetyl groups to histones, resulting in gene modification or activation and increased gene expression. And then there's histone deacetylases, or HDACs, which remove acetyl groups from histones, resulting in gene deactivation. So just imagine like genetic material wrapped around these histones that is either being, blocked from being used by these histone deacetylases, or it's being enabled to be used by acetyltransferases. So it's just like turning it on or off, depending on which enzymes are at work for that on that particular histone. So this histone thing, these histones, they are all over the DNA. You can find them all across the DNA because they're necessary for just packaging the DNA into your cells. So if you look at the genome, you can kind of put something on top of it. There's like another layer on top of it that you could refer to as the histone code. So like, take this for example. This down here, this is your genome, and right above it, and as you can see, blocking parts of it is the histone code. The arrangement and structure of histones are influencing the way DNA is read and transcribed. So if your genetic code is like a blueprint on how to build something, the histone code would metaphorically be like the paper that blueprint is written on. So even if you have like the best plans for building something, if you don't have the right kind of paper, you're gonna have trouble representing those plans the way you intended. So it's all about like the shape and the size and things like that. Histone code can have these major effects. Now, related topic when it comes to all this stuff about genomes and epigenomes is we've done a series of studies called genome-wide association studies that involve scanning the genomes and epigenomes of a wide range of individuals for specific variants that are believed to be responsible for specific traits. Like we're basically looking for a significant difference in a trait between people who have the variant and don't. So this is just one way of figuring out like how do we read the genome and epigenome and try to make some accurate predictions. And these kinds of studies, these genome-wide association studies, have definitely helped us to better understand how these epigenetic mechanisms like methylation and histone acetylation can influence a wide range of physiological and psychological traits. Now, like I was saying earlier, like the methylation and these histones, like these kinds of things can be inherited. So you might have some modification to your epigenome that then prevents certain segments of your DNA from having an effect. And that modification that happens during your lifetime could be inherited by your children. So we know there are a lot of things like this that could be inherited. Like we know that there are predispositions that you could gain, such as autism or cancer or heart disease or diabetes or learning and memory issues, lifespan, physical aggressiveness, and many, many others. Okay? Like there's a lot of things that could modify your genetics at this epigenetic level that could then get passed on to your children. But a lot of people, they just don't really know much about this. Like personally, I didn't really know much at all about epigenetics until I started teaching this course, right? Like this is really new material. This is really cutting edge kind of stuff. So we're learning a lot. This is all really new and cool research we're learning here. And as we learn more about this stuff, we're definitely learning a lot more about what we can do with it. Like we know, for example, that exposure to sex hormones can have a large impact on DNA methylation. During early development, certain methylated sex genes can result in like a marked incongruence between gender identity and physiological sex characteristics. And we've done long-term studies on things like gender-affirming hormone therapy that have shown that this actually results in a decrease in methylation for those receiving feminizing treatments and an increase in methylation for those receiving masculinizing treatments. So whether we understand it or not, we are modifying the epigenome by providing these kinds of treatments. And what this seems to show is that, like other traits, things like sex traits can be modified through these kinds of environmental epigenetic forces. So the whole point here is that your genome can be modified and your children can inherit it. And most of that modification is going to be happening early in life. Your early childhood experiences is when you're most susceptible to this epigenetic force, this Lamarckian inheritance, if you want to call it that. And there's a wide range of things that are very likely to cause epigenetic change, like stressful experiences early in life, like traumatic experience kind of stuff, like just having a childhood where you are put under a lot of pressure is likely going to cause some kind of adaptation in this way. So this is, in many ways, exactly what Lamarck was talking about. This is like a rapid form of evolution. And it's like I'm saying, environment can be all kinds of things. It's not just about your surroundings. It's also about the kinds of foods you eat. Like the food you eat when you're very young can modify your epigenome. What your mother ate while you were still in the womb, that can also modify your epigenome. Like nutrition plays a big role in this. But yeah, generally when we do talk about like epigenetics, we are talking about things that are potentially more unpleasant, like stressful early life experiences, like childhood maltreatment kind of stuff, right? Like in one particular study, we took rats, like baby rats, rat pups, and these rat pups received low levels of maternal licking and grooming during their first week of life, so they just didn't get as much affection from their mother. These rats showed a decreased histone acetylation and increased DNA methylation in areas related to the stress response. So simply not getting enough affection from their mother caused these rats to have a modification to how their DNA is going to be read for the rest of their lives. Big influence, lifelong influence for these rats. And it works the exact same way for people. So we know a few things, obviously, but we've still got a lot to learn about how the epigenome and genome combine. We sometimes call this combination of two things the nucleosome, but that's not important. What's important is we're learning. Every day we're learning more about how the epigenome works, how it modifies the genome, and how we can modify the epigenome. Like we're gaining more and more control over how DNA is read and expressed. And this is leading to a lot of promise, like therapeutic interventions, like treatments for things that we never even thought we could have a treatment for before, right? So what is very clear from all of this research in epigenetics is that the processes underlying development and behavior are a lot more complex than anyone previously imagined.